Hey, Carson, Stella, Rachel. Glad you could all make it today. All right, Kate. Not Rachel. All right, wonderful. How are you all? Reagan, I love the color of your walls. That is so bright. Did you paint it yourself?
My mom did it.
It probably took like 10 coats to get that vibrant color, right?
I think it was only 2.
Really? That's pretty good. That's
Yeah.
awesome. Awesome. All right, anybody have any questions before we get started? All right, let's dig right in. All right, let me know if you cannot see that. Beautiful biology is life. Because of course it is. All right. All right, so before we get started, I will remind you guys of this again next week, but 2 weeks from today, we will not have our live class on Monday. I will be out of town. I don't like canceling class. I really try not to do it, but every so often something happens that is just unavoidable. So this was one of them. Now you guys can come to the Wednesday class if you want to. The Wednesday class meets, it meets at 3 o'clock Eastern time. You can come to that same link. We all use the same link. Or if you can't come to the Wednesday class, you can watch the recording that I will post before the end of the day on Wednesday. So you'll be able to watch that recording. All right. Like I said, I will send you a reminder. We'll talk about it next week. And then I will also send a reminder the day of that Monday to remind you there's no class. Okay. Now I want to talk about the osmosis lab. Isn't that cute? Wouldn't it be nice if we could learn through osmosis like that? Just lay on a book and from the high concentration of knowledge to the low concentration of knowledge, things would move. But I want to talk about our lab, the egg lab that we did. So egg cells are usually the largest cell that an organism makes. Remember, an egg is one cell. So it's the largest one because the egg contains the organelles, the energy sources, other cell parts that are necessary for the development of the organism. The sperm cell contributes only DNA during fertilization. All the organelles, all the other parts of the cell are in the egg. Now, bird eggs, like the chicken egg that we did, are some of the largest single cells on Earth. Once they're fertilized, that cell very quickly begins dividing into billions of cells that then make up the tissues and the organs and the organ systems of that new organism before finally hatching in the case of an egg. Now, in this lab, we used a chicken egg, and we wanted it to give us a model to see how an egg is affected by its surrounding environment. So the very first thing that we did is we put the egg in a solution of vinegar and we let it sit for a couple days. Why did we do that? What was the point of the vinegar? Kate?
To remove a lot of the shell?
Yeah, we wanted to dissolve away the shell. So chicken eggs are made out of a chemical called calcium carbonate. It's actually the same material that's found in seashells. Same thing. And the vinegar is acidic, so it will dissolve away that calcium carbonate. Okay, so what happened to the egg once the shell was gone? Once that shell was gone, still sitting in vinegar, how did the egg itself change? Ella?
I can't see you. It became translucent.
It did. It became translucent. Most of them became translucent. We'll talk about why a couple of them didn't in a minute. What happened to the size of the egg once that shell was gone and it was translucent?
It got big.
Yeah, it got a lot bigger, didn't it? Why?
Can I be honest?
Yeah.
Oh wait, actually
Always.
I think I know the answer. Because the liquid like made it expand. Like
what liquid?
the vinegar.
Yeah. So
Okay.
as osmosis happened here. Remember, once we removed the shell, now we're left with the membrane, just the outer membrane of that egg. and the water was able to move through the membrane because the membrane is permeable to water. It lets water move both ways. But remember what osmosis is. It's water moving to an area to try to spread out the particles. So it's moving from where there aren't as many particles to where there are a lot of particles. And that can happen back and forth across the egg. So basically the concentration of particles inside the egg was greater than the concentration of particles in the vinegar. So water moved into the egg through that membrane to try to spread out those particles. And the egg was able to get bigger, so it did. That's osmosis, part one of osmosis. Now, the next thing we did after our egg was big and translucent, we poured out the vinegar and we covered the egg with Karo syrup. What happened after a couple days? How did the egg change in the Karo syrup? Reagan.
It shrank.
Yeah, it shrank. So why did it shrink? Same reason it got big in the first place. It shrank because it was osmosis, just in the opposite direction. Now that corn syrup was very concentrated. Corn syrup's mostly sugar and it's highly concentrated sugar. So it had a higher concentration of particles than the egg did. So now water's going to move out of the egg to try to spread out the sugar particles that are in that Karo syrup, right? It's trying to reach equilibrium. Basically what it's trying to do is have the same concentration of particles on both sides of that membrane. Well, when water moved out of the egg got smaller. It shrank, it shriveled up a little bit. Now, there were a couple of you who wrote to me to tell me that your eggshell didn't seem to be dissolving. And interestingly, all of the non-dissolving shells were brown eggs. So I wanted to know, do brown eggs make a difference? So I decided to run an experiment of my own so I could see if there was a difference in the way that brown and white eggshells behave in this experiment. All right, now this is what my eggs looked like at the very beginning, before they were ever put into the vinegar. First of all, do any of you guys have any idea why some eggs are brown and some are white? What makes them that way? It has all to do with the breed of the hen. White eggs come from breeds that have white feathers, usually, and brown eggs come from breeds that have brown feathers. Simple as that. All right, so I took my eggs, the white one and the brown one, and I put them down into a jar and I filled it with vinegar. And immediately I saw bubbles forming. So I took this little video so you can see the bubbles on both of them. So far, so good. So far, they're both behaving the same way. Now, vinegar dissolves those eggshells because it has a compound in it called acetic acid. And that acetic acid reacts with the calcium carbonate in the shell, right? It makes carbon dioxide gas and water. That's what you're seeing bubble up, are the carbon dioxide gases. So you can observe the reaction happening as the carbon dioxide is going up. The water is going, just mixing in with the vinegar, so you don't see it. All right, now after it had sat in vinegar for 2 days, I looked at them. They looked like this. Now, both of those shells were dissolved away. But when you look at it, it doesn't look like the brown one was dissolved. The reason is because the membrane itself is brown. So the brown egg did not become translucent like the white egg did. But because the shell was gone, it did allow water to move through the egg, to move into the egg through osmosis. So if we compare them, if we look at the size here of this white one before and then here after, you can see a bit of the spoon around the outside of both of these. It's much, much wider, much larger here. Both of them are much larger. So notice how much bigger both of those eggs are after we put them in the vinegar. And they were both soft. There was no shell left, and they resisted pressure that I applied with my fingers. When I tried to squeeze them, they kind of felt a little bit like a filled water balloon. Like I could push in a little bit, but I could tell they were full. But there was no difference between those two eggs. There was no difference in the way they felt. They look different though. The brown one still looks like it has a shell. The white one is translucent and clearly doesn't have a shell. So then I poured off the vinegar, I added the Karo syrup, and I left it for 24 hours. And this is what the eggs look like. Definitely you can see they've shrunk. This one hasn't shrunk nearly as much though as the white one did. Now this is the way that they were, how soft they were. So watch this. You can see it's kind of squishy. But this one is much squishier. Much squishier. So you can see the way the white egg really shrank. While the brown egg was soft, but it didn't lose as much water as the white one did. Anybody have any thoughts why? Why maybe the shell's gone? Why maybe did the brown one not lose as much water in osmosis? What are some possibilities? Well, I went to the research. I went to the internet and I did some reading to try to figure out why. So here's what I found out. First of all, eggs of the same size. There was an experiment that somebody ran where they compared to different kinds of eggs. They compared white eggs and brown eggs. They used the same size eggs, but the brown ones weighed more. Same size, more weight. Why? Why would the brown egg weigh more than the white egg did? What makes something weigh more? It's going to have more particles. Something weighs more if it's more dense, right? Just like a, you know, if you've got a balloon full of feathers and a balloon full of sand, they're both full, but the sand particles are much denser. So that balloon's going to weigh more than the feathers do. So the egg weight was greater for the brown eggs because a brown shell is thicker than a white shell is. Now, that probably means that it's going to take longer for that shell to dissolve away. All I know is they were both dissolved away in 2 days. 48 hours, they were both gone. Now, another thing that they found, difference between white and brown eggs, is related to what's called static compression strength. Brown eggs had greater static compression shell strength than white. So think about that for a minute. What do you think static compression shell strength is? Think about all those words: static compression shell strength. Toby.
I guess how like durable it may be, or
Durable to what? What does compression mean?
to
Right.
like, you know, like forces trying to like go inside.
Yeah, exactly. Forces trying to crack the shell, basically. So this is the test that they do. This is the test for static compression shell strength. So eggs are they're just a whole bunch of roundness, right? They're all oval and there's no square flat edges. Things that are rounded tend to have much greater compression strength. That's why we use rounded structures so often in things like buildings. To distribute the load across a curved surface is much more effective. So airplanes are curved and buildings have curves in them, and other design engineering structures will have curves. So it definitely, the brown egg, it was able to take more pressure than the white one. Okay, that's another difference that we have right there. Now, a vitelline membrane is what we call the membrane between the yolk and the white. So it surrounds the yolk, protecting it, separating it from the white. So it's stronger in a brown egg. Do you guys think that that vitelline membrane is significant for the experiment we just ran? Let me tell you, it's not. Why isn't it? Why don't we care what's going on with the vitelline membrane? Ben. You're muted, Ben.
Is the shell itself that's more important? So that's in compression.
Right. We're not really concerned what's going on inside the yolk. We're concerned what's going on in the white, in the albumin on the outside. We're more concerned with the shell membranes. So there actually are two shell membranes. If you look here, you can see the inner membrane, which is close to the white of the egg. And then there's the outer shell membrane, which is closer to the shell of the membrane. Now they give that physical barrier. They really do a good job of protecting that egg from really bacterial infection. we don't want bacterial to get in there. Now, once the eggs fertilize, they play a really important role in regulating oxygen and carbon dioxide moving back and forth across the shell. So when they were tested, there was no difference between the shell membranes of a white egg and a brown egg. That's interesting to me. So we learned a couple things. We learned that the shell in a brown egg is thicker, and that would impact the speed that it dissolved in the vinegar. Once that shell was gone, though, since there is no difference in the composition of those shell membranes or of the egg white itself, they're all albumin, we would have expected they would behave the same way, but they didn't. For some reason, there was less movement of water by osmosis out of the egg and into the Karo syrup in the brown egg. Okay, so really, the mystery continues. I can't tell you why the brown eggs didn't work. I haven't found an answer to that. What I did learn is that I will tell future classes to use white eggs. I'll tell everybody, don't use a brown egg because it may not work. But for those of you who did use a brown egg and said it didn't dissolve away the shell, it did. It just doesn't look like it did. You still could continue on with the experiment. And one thing that I did notice is that when I poured off the Karo syrup to get the eggs out, the Karo syrup that had the white egg in it was much more watery. And that makes sense. Water came out of the egg and watered down the Karo syrup, where the one that the brown egg was in was still really thick, really kind of, you know, very viscous. Kate, did you have a question?
Does the size of the yolk change at all when the egg is getting bigger or smaller?
It didn't seem to. That's a good question. And I noticed that, and I tried to, it's kind of hard to tell what size the yolk is when there's so much water in there. But as I tried to look at it, it didn't look like it changed. And when it shrank down, so let me go back to this page here. When it shrank down, here we go. So you can't see the yolk in this one at all. But when we pick this up and it shrank down, now I can see the yolk, and it looks to be about the same size that it was when it went in there. So I don't think there was any osmosis back and forth across the vitelline membrane. Yeah, excellent question. Toby, that happened to you with the brown egg? Yeah, so it didn't look like it was dissolving? Well, that's why. It was, though. So you could continue this experiment, but, you know, in the future, we'll just use white eggs. All right. Anybody have any questions about that? That was fun. I love it when you guys ask me questions that make me do research. I really love it when you ask me questions that lead to new experiments. So what could we do to further this experiment? Run it more times. That's Toby. Is that what you're going to say?
I like use more brands of eggs.
Yep. I could use different brands of eggs. I could use different brands of that, you know, the syrup, the Karo syrup. Try with green eggs, like green eggs and ham. One of my favorite Dr. Seuss books is green. Yeah, eggs come in different colors. Eggshells come in different colors. That would be a really fascinating experiment, to get a bunch of different eggshell colors and try that experiment that way. I could also try pouring out the Karo syrup after a day and then putting new Karo syrup in, and see if it just maybe the same effect would happen with the brown egg. It just might take a little longer. And I don't know why. I cannot find, I spent a couple hours looking, trying to find an answer to this, and really didn't find one. There's no difference in those membranes. So once the shell was gone, they should have behaved the same. Kate.
You could continue to try and find differences between both of the types of eggs, like maybe the one has something slightly different inside it that would make the water not go across as much.
Yep. It doesn't seem as though there is. But again, just because we haven't found something doesn't mean it's not there. So everything that I read said that the white is exactly the same between two different kinds of eggs, and the membranes are exactly the same. The shell is where the difference is. And the biggest difference is the color. Yeah. Now, there are other possibilities. Eggs are different depending on what the chickens are fed. So if chickens are fed a more natural diet, the yolks are going to be almost an orangey color, where if they're fed a vegetarian or a like feedlot kind of diet, the yolks are going to be very pale. So I think that there could be differences there too. So what I should do is make sure that both eggs either are from a feedlot or both eggs are from a free range, organic chicken farmer, something like that. But this is a sample size of 2. I had 1 of each. I could run this with 10 and try to see what was different. All right, let's move on now to our experiment. So we talked about photosynthesis last week, and you guys, this week, you're going to learn about a process that's in a lot of ways the opposite of photosynthesis. Cellular respiration is sort of photosynthesis backward. Hopefully you were able to get the supplies to participate in the lab today. If you weren't, it's okay to observe, but you all need to complete that lab sheet that I sent you this morning. So if you didn't check your messages this morning, there's one there. Please check every morning before our class, because I will tell you the things that you need. And there's a lab sheet there that you'll need to complete because we'll talk about it next week. Okay, just as a reminder, a real quick review. What are the reactants of photosynthesis? Remember, reactants react, they go in. What are the reactants in photosynthesis? There are 2 things plants take in that they need in order to perform photosynthesis, other than that light energy. Toby.
Oxygen and water, right?
Not oxygen. What do plants take in? They release oxygen. Stella. Stella A.
Carbon dioxide.
Yeah, carbon dioxide. Carbon dioxide. So they take in carbon dioxide, they release oxygen. So oxygen is one of the products. The reactants are CO2 and water. The products are sugar, usually glucose, and oxygen. There are other sugars that are made in photosynthesis too, but the one that we focus on for our learning is glucose because it's the most common one. All right, now energy is locked up in that glucose molecule. We have to get that energy out of the glucose molecule. That's what respiration is about. That's what you're going to learn this week. All right, so remember there's 2 stages of photosynthesis. Anybody remember what stage 1 is called? There, go ahead, Toby.
The Calvin cycle?
No, that's stage 2. Good guess though. I love it when you answer. When you're trying. And you're right, it's one of the stages. It is one. All right, so the first one is called the light reactions. So they're called that because they need light. So it's called either the light reaction or the light dependent reaction. The second one is called the Calvin cycle. Sometimes you'll see the Calvin cycle called the dark reaction or the light independent reaction. Now remember, the whole thing needs light, because even though the Calvin cycle itself doesn't require light, the light reaction does, and the Calvin cycle can't happen without the light reaction. All right, so what comes out? Sugar and oxygen right here. Oxygen is released in the light reaction. Oxygen is just a waste product. The cell doesn't need it. The plant cell doesn't need it, so it just gets rid of it. Fortunately for us, because it's what we need to breathe. We have this great interaction between us and plants. Animals that breathe oxygen and plants cooperate with one another. They give us oxygen, we give them carbon dioxide. Perfect. All right. Now, what went too far? All right, here we go. All right, so if you're ready, we're going to do our experiment now. And this is going to show you the importance of light in the light reaction. All right, so this is what your lab sheet looks like. Get it out or open it on your computer, whatever you need to do. When we're doing any kind of a lab, the very first thing we're going to do is read the whole thing. You always want to read all your materials and all of your instructions. So I'm just going to real quickly read through this. All right, what's our objective? To observe how plants utilize carbon dioxide and release oxygen during photosynthesis by using bromothymol blue, a pH indicator, to detect changes in carbon dioxide concentration. That's what we're trying to do. That's what we're trying to do here. In a nutshell, we're trying to see how photosynthesis releases oxygen into the water that the plant's going to be in. So here's our background information. Bromothymol blue, this little bottle of stuff that you got, is what's called a pH indicator. it changes color depending on how much carbon dioxide there is. So in neutral conditions, just plain neutral, like the first thing that what we'll do is put it in water. That's a neutral situation. This is going to be blue. But as carbon dioxide dissolves in water, it forms what's called carbonic acid, and that lowers the pH, turning the solution yellow or green. During photosynthesis, plants take in CO2 from the environment to produce oxygen and glucose, which can cause a decrease in the amount of carbon dioxide in the surrounding solution. Okay, so if carbon dioxide turns it yellow-green, taking carbon dioxide out is going to revert it back to blue. Does that make sense? Oxygen makes it blue, carbon dioxide makes it yellow. Put in more oxygen, it becomes more blue. Put in more CO2, it becomes more yellow. Take out oxygen, it becomes more green. Take out CO2, it's going to become more, or take out oxygen, it's going to become more yellow. All right, now, what is pH? This whole thing's about pH. What is pH? What do I mean by that? Toby.
The level of acidity in water.
Yep, the level of acidity on one end and alkalinity in the other end of any substance, not just water, all of them. So all substances have some kind of pH to them. They're either going to have a low pH and be an acid, or they're going to have a high pH and be a base, or they're going to be a neutral, which is around 7. So this chart shows the different pHs of different substances. Now, something has a high pH when it is alkaline. Sometimes we say basic. So bases or alkalines, they have a high pH because they have more hydroxide ions in them. Those hydroxide ions make them less acidic. And the farther they are along the pH scale, the more hydroxide ions they have. Something has a low pH when it is acidic. Acids have a low pH because they have more hydrogen atoms in them. All right, more hydrogen and less hydroxide. Now, the way I remember this, which is hydrogen, which is hydroxide, for me, I think about hydrogen on the periodic table. So hydrogen is the smallest of all the elements. So I remember that the smallest pH numbers have more hydrogen. The smallest pH have more of the smallest element. They have more hydrogen right here. Small pH, lots of the small atom, those hydrogens. All right. The color of bromothymol blue at different pHs can be shown here. Now, this is when you put 1 drop in. This is when you put 2 drops in. So if the solution is 5.5 pH on the pH scale, this is the color it's going to be, all the way up to about an 8. That's about the range that bromothymol blue has. That's just for your interest. You don't need to tell me pHs of this. All right, now back to our chart here, our worksheet. So the next thing we're going to do is look at our materials list. Always read through and make sure you have all your materials. If you find that you don't have water, you forgot to get water, you have to get up and go get water, right? So you need to always make sure you have all of the things there. I'm going to assume that you do. Now, the next thing is going to be our hypothesis. So we always want to think of a hypothesis before we run an experiment. Let's try to think of one together. What do you think will happen to the color of the bromothymol blue solution when the plant undergoes photosynthesis in the light? What will happen in the dark? All right. First thing we want to do is know what we're going to do. What's our independent variable? Independent variable is the one you change, right? What are we going to change here? Kate?
The amount of light.
Amount of light, and we actually have 2 variables here that we're going to change. We're also putting a plant in. So we're going to put a plant in one and not in the other. So we're going to change the amount of light. We're going to add a plant. So this really might be a 2-part hypothesis. We'll talk about that. What is our dependent variable? What are we going to measure or look for? What do we expect is going to happen as a result of this experiment? Toby?
The color of the solution.
Yep, exactly. That's our dependent variable. So now we can put that in if-then format. The then is going to be the color of the water, a color change in that water. The if is going to be what we're doing, our independent variable, the light and the plant. So our hypothesis might be, well, you guys tell me, somebody give me an if-then that could be good for this. I know it can be kind of intimidating when I throw these kind of questions out there. Let me just tell you, that's what learning is all about. You know, the whole learning process is finding out what you don't know and then learning more. So we learn more when we get things wrong. I do anyway. I learn more when I get something wrong than I do when I get it right. So Reagan, give it a shot.
If the plant and bromothyle solution is placed in the light, then the solution will remain blue.
Very good. Very good. Or not necessarily remain blue, because you'll find out in a minute, we're going to change its color. So we're going to change the color and it will revert back to blue. That might be a good way to put it. So we could also do it with the dark one. If a plant is placed in a bromothymol blue solution and then placed in the darkness or kept from light, then the color of the solution won't change. So we could have a two-part hypothesis there. All right, next thing we're going to do is read through our procedure all the way through before we start, always, always. So I'm going to read it real quickly. Put on safety goggles. Prepare the bromothymol blue solution. Put 150 milliliters, about 5 ounces, that's a little more than half a cup, of water into a 2-cup measuring cup. Add 5 to 10 drops, about a teaspoon of bromothymol blue. You need to keep adding until the solution turns blue. We'll talk through that. You're going to stir until it turns blue and observe the color change. Number 3, create a carbon dioxide rich solution. Gently blow into the BTB solution using a straw for about 30 seconds. That adds carbon dioxide, making the solution greenish yellow. It makes it more acidic. It moves it down on that pH scale. Number 4, set up your control and experimental groups. Pour that carbon dioxide rich bromothymol blue solution into 3 test tubes or jars. Label 1 as control, 1 as experiment light, and 1 as experimental dark. Experimental setup. Place a plant in both of the experimental test tubes and seal them with a stopper or lid. Leave the control test tube without a plant. Seal it with a stopper or lid. Light and dark conditions. Wrap the experimental dark test tube in aluminum foil to block the light. Place all 3 test tubes under a light source. Record your initial observations. On the chart below, note the initial color of the bromothymol blue in all 3 test tubes and observe over time. On the chart below, record any color changes at intervals of 15 minutes, 30 minutes, 1 hour, 8 hours, and 24 hours. Now we'll do these first ones, the initial and the 15 and 30 minutes together. You guys will have to do the 1 hour, 8 hour, and 24 hours on your own. Okay, then you're going to put them all on this chart. That's where we're going to collect our data. All right, let's go back to this experiment and now we'll begin. So the first thing you do, put on your safety goggles. This is important because if you get any of this on your skin, it can be an irritant. If you get any of it in your eye, it can really irritate your eyes. Not going to cause you to go blind, right? It's not that toxic, but it could be an eye irritant. So we put on our safety goggles. Aren't they lovely? Fashion, fashion. All right, is anybody doing this experiment? Okay, good, good. So if you don't have safety goggles, I highly recommend getting some because it's, you know, some of the experiments that we'll do, it's a good idea to have. If you don't this time, just be real careful. Just be real, real careful. Okay. I'm cleaning mine because they're so dirty I can't see through them. There we go. All right. Much better. Okay. The next thing we're going to do is prepare our bromothymol blue solution. So I have here 150 milliliters of water. I have mine in a flask. This is called, yeah, you can just wear your glasses. That's fine. You can put them in a Erlenmeyer flask. Now, let me say, sometimes you can just wear glasses. Sometimes you absolutely have to have a safety goggles. So in this one, it's not, most of what we're going to do here in our class is not going to be so dangerous, but you know, it's always better to have glasses. So I've got 150 milliliters, which is a little more than a half a cup. So if you don't have anything that measures milliliters, that's fine. You can just use the cup measure and fill it up, you know, like a half a cup and then a splash more. The amount doesn't necessarily, it's not exact. You can see there, the next thing we're going to do is add 5 to 10 drops of that bromothymol blue. All right, I'm going to tip my camera down so you guys can see what I'm doing here. Let's move that back out of the way. And there we go. All right, so now I'm going to take this bromothymol blue solution and I'm going to add, that's not great. Okay, yeah, that's okay. And I'm going to add enough drops that my solution turns blue. So anywhere between 5 and 10, you might need to add a little bit more. Okay, there's 1, 2, 3, 4, 5, and I'm going to swirl it. If you have a measuring cup and you don't want to swirl it, you can use your straw and the opposite end of the one you're going to drink. Now that is blue, but I want more. I want a darker blue solution. So I'm going to go up to 10. 1, 2, 3, 4, 5 more. That's a little bit darker blue. I'm actually going to go a little bit more. I'm going to put 5 more. So if you want to go, the more drops you put in, the darker your blue is going to get up to a certain point. Okay, that's probably about as blue as I'm going to get it. So you can see that kind of teal blue color. Okay. Now observe the color change. So those of you who are doing it, observe your own. Those of you who are not doing it, observe mine, and you're going to want to write that down when we get to that point on the chart. I'll walk you through it. Okay. Now the next step, create a carbon dioxide rich solution. Gently, you don't want to splash this all over. So especially if you've got a wide open measuring cup, you don't want to blow to where you overflow your jar. So you want to gently blow into the bromothymol blue solution using a straw for about 30 seconds. This adds carbon dioxide, making the solution greenish yellow. Now, depending on how much carbon dioxide you have in your breath, it may take a little bit longer or it may happen faster. You just want to keep blowing until you see a color change. All right, so you guys can watch as I blow into my solution and watch for the color change. I'm actually going to make this, hold on, I'm going to stop this share. And make this bigger. Okay, so you can see a little better. So I'm starting to see a color change. It's not quite that bright, vibrant blue that it was before, but it's not enough. Let me make it more. Okay, you guys can see the difference now. It was that bright blue before. Now it's this pale kind of greenish yellow color. One more good for just first to do it. Okay, there we have a nice pale green color, pale yellowish green. All right, so that is my carbon dioxide rich bromothymol blue. Now what I'm going to do is get my test tubes. So I've got 3 test tubes here and I labeled them control, experiment light, and experiment dark. I don't know if you can see that, EXP dark. I don't think I told you guys to bring a marker. If you don't have a marker, you can just do it on a piece of paper and then, you know, set them by your paper. So I'm going to pour 1/3 of my solution into each of these 3 test tubes. All right, so I'm going to pour there. You can really see how pale the color of that is when you look at that. I'm going to have about the same amount in each tube. So I'm starting with 35 milliliters. I have a little bit more, so I'll go a little more into each of these. Doesn't matter if it's exactly, just so it's close. That's going to be a little too much. You want to leave a room for your plant. So I'm going to pour some of this off to leave a little bit of room for my plant in that. Okay. Now set aside your other, you know, whatever's left. If there's any left in your jar or measuring cup, put that aside. All right, now I'm going to place a plant, and I have my water plant right here. These are the water plants that I have. And I'm going to place one piece of the plant into each of the solutions. So I want it to be about the size of the test tube. So I'm going to cut it off so that the whole thing will be able to go down inside. And I'm going to put one in one of them and cap it with a lid. I'm going to put one in another one, get another piece of the plant. Break it off so that the whole thing will go down in there. Push it down in and put the lid on that one. And then the third one, I'm not going to put a plant in. That's my control. I'm just going to put it on right there. Whoops. Now, I will tell you guys, we will do another experiment later this year, at least one, maybe two, with bromothymol blue. So hold on to it if you bought it, and consider buying some if you didn't. Because I think it's better to do experiments than to watch them. Okay, so there's our three right there. I'm drying them off. So I've got my dark, my light, and my control. Now I'm going to take my piece of foil. I'm on number 6 now, light and dark conditions. I'm going to take this foil and wrap the experimental dark in foil. So I'm picking up the one that says experimental dark, and I'm going to wrap it in foil, and that will keep any light from being able to get at that test tube. Now I have way more foil than I need, so I'll break off. I don't need to have that much. Okay, I'm going to wrap it up like that, seal the ends. I'm preventing any light from getting to this plant. So think about what I'm doing to the light dependent reaction. The light dependent reaction requires light. That's also the phase in which oxygen is given off. So something's going to happen there. All right. Now I'm going to place all 3 of these test tubes under a light source. That is part 2 of number 6. Let me put this back up. Okay. All right. This experiment is ready to go. So now I can put it under a light source. If you have a window that's nice and sunny, you can put them there. If you don't, you can just put them under any, it doesn't have to be like a grow light or anything. You can just put them under any light. Put it in a bathroom and leave the light on overnight. It's important that you have 24 hours worth of light. Okay, now we're going to look here at this chart. Let me share my screen again with you guys. Okay, everybody can see the screen? Okay, now, if you got any of this on your skin, please use your wet paper towel to wipe it off. If you got a lot on your skin, go wash your hands now. If you splashed it all over you, just go wash. Okay, now let's look at our data sheet. Remember, when you're submitting a lab report that asks for data and only data, you could use this sheet. We're not writing a lab report on this one, but we could and use just this sheet. Okay, so the initial observation. What color was our control in the initial observation right now? At the very beginning, what color is the control? Like yellowish green. So you'd write yellowish green on your the first space, initial color of control, no plant. Color of experimental light with the plant, it is also yellowish green. And same with the dark one. So for all 3 of those, it's going to be yellowish green. All right. Alexa, set timer for 15 minutes. 15 minutes. All right. In 15 minutes, we'll come back to these. So just set them aside. Let's have a little discussion while we're waiting. And then we'll come back again after 30 minutes. And then, obviously there won't be enough time to do the whole thing, to do an hour. So you'll have to do the hour, the 8 hours, and the 24 on your own. Those of you who are not doing the experiment, you will just leave those blank, and I will tell you when we meet next time what happened, what to write. And you know what? Actually, I'm going to do. Tomorrow morning. I'm going to send you pictures. Okay, I'll send everybody pictures so you can complete your chart and you can see what's happening and you can think through some of this, because I do want you to finish the whole lab sheet and there's some analysis that you'll have to do based on the pictures. So first thing in the morning, after 24 hours have gone, well, it won't be 24 hours until 4 o'clock tomorrow. So as soon as 24 hours has gone by, I will send it to you this time tomorrow. Okay, so let's talk about a couple things. So the color of the original bromothymol blue in water, what was that? What color, when I first put the bromothymol blue in the water, what color was it? Ben?
Really, really blue.
Yeah, it was a really nice blue color. Why did I put on goggles? Toby?
Could have caused like irritation.
Yeah, safety first. Safety first is the rule in any science experiment, in any lab that you're in anywhere. Safety first. So once again, if you got some on your hands, go wipe it off. If you got any on your face or your eyes, flush out your eyes. All right. Why did I make a control test tube? Remember, the control is the one, it's this one. I blew the carbon dioxide in, but I didn't put a plant in. Why? What's the point of this? Ben?
If you don't have a reference point, then you can't really judge what's happening with the
Yeah.
bottle.
Yep, I need that ability to compare. Your reference point is a good way to look at it. So I'll compare the two tomorrow, well, throughout the experimental times. Yep. Why did the color change to greenish yellow when I blew into it? What was I putting in it that made it change color? Kate?
Carbon dioxide.
Yep. What did carbon dioxide do to it? I'll make it more specific. What did it do to the pH? Ben?
I lowered it.
It lowered the pH. It made it more acidic. This is the big reason that we breathe out carbon dioxide. Carbon dioxide is very acidic and it will very quickly do damage to your body if you don't have a way to get rid of it. All you have to do sometime is try holding your breath. And it doesn't take very long before you start feeling lightheaded, before you start desperately needing to breathe. That ability, that need to get carbon dioxide out is so powerful that if, like, you know how kids sometimes they get so mad they hold their breath, they like pitch a little tantrum and they hold their breath, their bodies will not let them die. It makes them unconscious, so they'll pass out and start to breathe unconsciously, because getting rid of that carbon dioxide is so important. You have carbon dioxide sensors all over your blood to test for high carbon dioxide levels because it is so acidic. All right, excellent. So why do I use a light source? Why am I going to put these in the light? Kate?
If you didn't use a light source, there wouldn't be any point in making one, so light wouldn't be able to get in.
Yeah. So what's the light going to do to the ones that it can get to? What's the light going to do to these? Anybody? Kate?
Probably photosynthesis will happen in the one with the plant. I'm not sure it'll do anything for the one without the plant.
Yeah, we expect nothing to happen in the one without the plant. Yeah, but we do expect that when we provide that plant with light, it goes into the light reaction, the one that depends on light. Stage 1 of photosynthesis is what is likely to happen. What comes off of a plant during photosynthesis? What does a plant release? Reagan?
Oxygen.
Yep, plant is going to release oxygen. And what color is bromothymol blue when there's oxygen present? Go ahead, Reagan.
Blue.
Yeah, it's going to be blue, that original one that I had. Now here's something interesting. I use a system called reverse osmosis water for my house, for my water. It takes everything out of water. When I tried this with water, with the bromothymol blue, it did not turn the solution blue. What that means is my reverse osmosis water didn't have any oxygen in it. Now, if I spun it around a little bit, if I swished it, I could add water to it, but it didn't because it turns blue when there's oxygen present. All right, so what color do you expect to see in this control test tube at the very end of this experiment? Kate,
Exactly what it is now.
I don't expect it to change. That's what I think is going to happen. What color do you expect to see in the tube that contains the plant and was wrapped in foil? Ben,
Well, I think without the photosynthesis, then there will be more CO2, so I think it's going to become paler, yellow.
okay. I think it's either going to stay the same or become, have more carbon dioxide in it. Now, the carbon dioxide's got to come from somewhere. Plants take carbon dioxide out of the air or out of the water. So I don't know that we have a source to add more CO2 to it, but that's a valid hypothesis that there might be. There might be, we don't know. But we know for sure it's not going to turn more blue, right? What color do you expect to see in the tube that contains the plant and is exposed to the light source? Kate.
Will it become more blue?
Yeah, it should become more blue in the presence of oxygen. So what if tomorrow I look at this and my control is blue? I'm expecting it not to change, but what if it does? What if it's blue? What can I assume? What happened? There was a flaw. Kate.
Either oxygen has been added to it or carbon dioxide has been taken away or both.
Yeah. So it's possible that my lid isn't tight enough fitting. If oxygen gets in there, like if I left this just sitting open, oxygen would get in there. Oxygen would diffuse right into the water and I would have a blue tube in the morning. So let's see here. How much time we got? Alexa, what's left on the timer?
You have 7 minutes left.
Oh, we're only halfway there. All right. So you guys are going to have to do all of the ones. Let me show you this. So we are going to, we're going to, hold on. Well, here, you're going to have to complete all but that initial one because we didn't even get to the 15-minute point. So like in 7 minutes, just after class is over, at the hour. So for me, it's 5 o'clock, whatever time it'd be on the hour for you guys, you'll check it. And then you'll check again in 15 more minutes and then an hour. So all of these are from the original time, 15 minutes from the initial, 30 minutes from the initial, 1 hour, 8 hours, 24 hours. I want you to complete all of these and I want you to fill out the rest of the chart. So there's an analysis section in there. I want you to answer those analysis questions. You don't have to turn them in, but I want them completed so that we can talk about them next week. You'll have answers to be able to give when I ask you questions. So whether you did the experiment or not, I expect you all to have completed your chart and completed your analysis of the chart of the results. Toby.
So I didn't do the experiment, so how would I know what it looked like?
I'm going to send you pictures tomorrow.
Thank
Yeah.
you.
At 4 o'clock tomorrow, I'll send pictures of all the different times and I'll tell you what they are. And you'll use that then to complete your chart and do your analysis. Yep. All right. Anybody have any questions? Kate.
For the experiment, were the plants kept somewhere light or somewhere dark? And would that affect it at all?
It shouldn't affect it unless they were dead. As long as there's still chlorophyll in those plants, meaning they're green, they should be able to perform photosynthesis. So they were kept in the light. They were kept in water, in the light, in my experiment. Yes.
To make it a controlled experiment, wouldn't we also need to have one without a planet that's being kept in the dark?
Probably. That probably would be a good, I try to make these experiments as simple as I can. Really what we should do is we should run 2 separate experiments, like one with the light and a control, one with the dark and a control. You know, both of them like that, but I'm just trying to simplify it here. But that's a very good point. Yeah. And, you know, you only want to change one variable at a time. We're changing two. So we are not really running a controlled experiment. It's still a very valid experiment though. Yeah. Yeah. All right. Anybody else? In 8 hours, it's going to be midnight for me. So how should I check the plant? Yeah. Just check it before you go to bed. That's a good question. Just whatever time you go to bed, check it and just make a note of that on your chart. That instead of 8 hours, it was 6 or 5, whatever. The big change is really going to be in the morning when you look at it after it's been 24 hours. Yeah. All right. Anybody have any other questions? I'll show you. Nothing's changed yet. So we're right about at the 15-minute point and we haven't had any change yet. All right. Time's up. Have a wonderful day. Enjoy the experiment. I will send it to you tomorrow. All right. Thanks for coming, everybody. Bye.